Hello viewers, welcome once again to our ongoing series on Mendelian genetics. Talking of the fifth part of what we call as beyond Mendelian inheritance, let us today focus our attention on another very interesting aspect of the interaction, which is the lethal genes. Till now, what we have studied is that the genes could be incompletely dominant as far as their manifestation is concerned, codominant, or they could be in a multiple allelic form, or what we read in the fourth episode was that they could even be pleiotropic, which means one gene could manifest itself in different types of unrelated or seemingly unrelated phenotypes. In one or two cases, we did find that in diseases it was a lethal combination. But then what in terms of lethality of a gene, whether it is going to be detrimental in a homozygous condition or a heterozygous condition or otherwise. Let us delve deeper into it. The lethal genes, in fact, were first discovered not very long ago, a century ago, in 1905 by a French scientist, Cuénot. Now, he was working on mice, and that too on the coat color of mice, that is in musculus. He recognized that there were two types of fur color, yellow and black. There was a color which was perhaps a blend of yellow and black hair. This was termed as Agouti, so the coat color. The black or the Agouti color, seemingly which was a blend, it was due to two recessive genes, that is capital A and capital A. On the other hand, the yellow colored mice were heterozygous. They were AY. Surprisingly, there was no homozygous AY which was appearing in the population. Very logical, because AY or AY is a lethal combination as far as the homozygous state is concerned. Cuénot didn't know that. All mature yellow mice have to be therefore heterozygous, that is AY. So, when he made a cross of these two mice, it was found that the combination of AY was a homozygous one, and it died in the embryonic stage itself. So, again the expected Mendelian ratio of 3:1 got disturbed, and it was modified to 1:2. So, what Cuénot had expected was that he expected the classical conventional expected 1:2:1 Mendelian ratio, but he always observed a 1:2 ratio, and therefore he was not able to produce any mice that they were homozygous. And how could he? Because they were in a lethal combination. So, one of the mutant gene of this Agouti character is going to result in another lighter yellowish color, which we have designated as AL. Now, when these yellow mice having the AL and AL, they are not lethal, they are again crossed with a homozygous wild type. Again, there is a ratio which is coming as 1:1 of yellow and dark gray. Now, this means that the yellow mutation is dominant, and all the parental yellow mice with heterozygotes of the mutant allele only would survive, and not the homozygous ones. Now, this whole problem was sorted out 5 years later by two scientists, Castle and Little, who confirmed Cuénot's work, and they further demonstrated that in a cross 25%, that is the one which we were talking about, 1:2:1, the last of the ones, that is 1/4 of the offspring, they were dying during the embryonic development. And therefore, this was in fact the first documented example of a recessive lethal allele. This brings us to a fundamental question: are the lethal genes always recessive? No. So therefore, we have different types of lethal genes. Recessive are one of them, is one of them. Then we have dominant, the conditional lethals, the balanced lethals, and gametic lethals, and some other subcategories. The recessive lethals, as the example of mice we have just observed, that the genes are going to be expressed only when they are in a homozygous condition. However, the survival of the heterozygote is not affected in this case. So, even in recessive lethals, we can have two categories. One, there would be no apparent morphological alteration in the heterozygote. In the second, heterozygote is surviving, not lethal, but then it assumes a distinctive phenotype which is different from the other one. So, an example of this would be in case of snapdragon, that is an enterhinum, and there are various human diseases also like cystic fibrosis, again the well established example of sickle cell anemia and androplasia. Just let us study one by one. A scientist E. Bauer in 1907, he first reported a lethal gene in enterhinum species, which is snapdragon, and he could observe three types of plants. Plants which were perfectly normal, having chlorophyll content, designated as CC capital, a pale green plant without chlorophyll. This was giving a golden hue, and it was a heterozygous one. So, he named it as an Aurea variety emanating from the sign for gold, which is aurum. And then the third category, so that means the second category was not totally white, but then they seem to be palish, pale green, more on the pale side. And the white plants, they were without chlorophyll. This was a homozygous recessive. So, the Aurea plants have the genotype CC heterozygous, as I said, and when two such Aurea plants were crossed, then the ratio, instead of the 1:2:1, came out to be different, and that is the two Aurea, one green and one white. But then in this case, the one white, which was one small c, was a lethal combination. So, again the ratio boiled down to a modified one, which is 1:2 in place of the expected 3:1. There is a similar example in Zea mays, where the gene controls chlorophyll production. It is a recessive allele G. It is going to control the synthesis of chlorophyll. However, if this small g is in a homozygous condition, it turns lethal. So, that means the kind of plants that we can have is only heterozygous capital G small g or homozygous capital G. So, in this case, interestingly, both of them are going to have the ones that survive are going to have similar phenotypes. So, a cross between two heterozygotes, they are perfect green, would give rise to one green, two that is 50% green, and one which is white. The one which is white dies. So, again the ratio again gets modified into 1:2. You could even call it as 2:1. Another very interesting example in human beings is the achondroplasia. You must have seen photographs of a population in various parts of the world where the entire family is of dwarfs and the body is distorted. This is in fact an autosomal dominant bone disorder, actually not a bone but a musculoskeletal disorder which causes dwarfism. The gene for this resides on the chromosome number 4. It is the FGFR3 gene, which is the fibroblast gene, and the inheritance of one of the endoplast allele, this is going to cause a disease. But if both of them are present, then it turns out to be lethal or fatal. So, on your screens you can see a cross between two individuals who are definitely affected, but then they are not dying, they are having physical deformities, and their marriage is again going to result in a situation which is a probability of 2:1 rather than the expected 3:1. Why? Because the capital A assumes a lethal combination. Brachydactyly is another character which exemplifies the lethal genes. This gene is located on the chromosome number 2, and that is on the longer arm, that is Q. And here you will find that the fingers of such individuals are unusually short, they are distorted. However, this type of an individual would be a heterozygote. This condition would turn lethal during the early years if the individual had a homozygous genotype, homozygous recessive, because there are going to be serious skeletal defects. Actually, what has happened is that a mutation has occurred in the IHH gene, which is the Indian hedgehog gene, it is popularly called, and this encodes for proteins which are responsible for bone growth and for differentiation. So, that means if there is a single copy of this mutated gene, then the phenotype would have few deformations of the skeletal bones. If both of them, so one single dose would be enough to make the protein for the other normal skeleton formation. However, if the genes are homozygous, then it turns fatal. Here is a picture of individuals who are affected by the disease. The other recessive lethal genes which are there fatal in human beings, there are many examples. 3 important ones are congenital ichthyosis, where also a child dies early, and the infantile amaurotic idiocy, where the brain is not developed much, and of course, the more famous and well known Cooley's anemia and the thalassemia of 2 types, namely major and minor. A discussion of alleles which are lethals and dominant ones is a little different, because in this case a fully dominant lethal allele is going to cause death even in a homozygous condition as well as in a heterozygous condition. So, the question now arises, if both the units are going to result in fatality, then how can these lethal alleles be transmitted from the next generation? They should be finished in one generation. Actually, the individuals with dominant lethal alleles are going to die before they can leave any progeny, that means in the early ages. In other words, there is a very poor degree of fitness. Therefore, this particular gene is automatically removed from the population in the same generation it arose. So, we will find that it is very difficult to detect a dominant lethal, because they are going to be immediately eliminated from the population. Let us see some subcategories of these dominant lethals, otherwise the dominant lethals would not be there in the population at all. So, that means we were talking about completely lethal genes, where the death would occur say in the zygotic stage, in the embryonic stage, or immediately after birth. So, that means not reaching the stage of sexual maturity, so that the fitness was very poor. Whereas, the other category is sublethal genes, which we also call as semilethal genes. Now, here you would find that the lethality does not occur in the juvenile stage. It occurs in fact in the sexually mature individuals. One classical example is of the epiloia gene. Now, this is going to cause mental defects, the various abnormal growths and tumors in case of heterozygotes, and a person may die before reaching adulthood. However, there are other examples, say in chickens we have the allele which is called as the creeper allele. The legs of such chickens is short and stunted, and by the way, creeper is a dominant gene. So, that means the heterozygous chickens are also going to display this phenotype, and when two creeper chickens are crossed, then the expected ratio from the Mendelian genetics would have definitely be 3:1. Instead, the ratio is now 2/3 creepers and 1/3 normal, which is again coming to 2:1. So, here is a display of the Punnett square, because the homozygous creepers, they are going to die, and in this case the ratio again gets modified. Very important example of the dominant lethality is in the form of Huntington's disease. Now, this gene resides on chromosome number 4, and it is a very pathetic neurological disorder in human beings, and it also reduces the life expectancy. However, since it is a progressive disease, the individuals who carry this allele would definitely reach a sexual maturity stage, and therefore, they can pass on this character to their offspring. Howsoever low is the fitness, yet the gene can be transmitted to the next generation, and this is how the allele gets maintained in the population. And there can be recurrent mutations also, which can now keep the dominant traits within the population. Huntington's disease is a very good example of what we call in molecular biology as repeated function. Now, repeated function is and there is an insertion also. In one of the places in a nucleotide, there is an insertion of the two triplets one by one, GAG and GAG, and the two amino acids of glutamine, they would come here, and this would alter the entire genome. In Huntington's case, if we have a father who is a heterozygous one, HH, and the mother is normal, that is having a homozygous recessive character. Now, in this case, we have these combinations which would give us a ratio of normal is to Huntington's in equal amounts, which is 1:1. There is a third category which is going to make these lethals express themselves only when certain conditions are fulfilled. So, such type of lethals genes are called as conditional lethals. In other words, they would require a definite or a specific condition to manifest themselves or manifest their lethality. There are so many examples in case of the mutants of barley, of zeamase, in case of neurospora, and even in Drosophila, where they are in fact temperature sensitive mutants. Each one of them would require a definite temperature, and this temperature is generally high to express their lethal action. So, that means if you are going to have an exposure to low temperature, then the lethality may not be manifested. Likewise, there is another mutant of barley, which is a chlorophyll mutant. Now, it would have a normal development of chlorophyll at say temperature ranges of 19 degrees or so, but then the moment the temperature goes below 8 degrees C, it turns fatal. So, here the situation is opposite. So, that means this is again a temperature mutant. Likewise, there are many other conditional lethins. They may require a particular type of light or a particular type of nutrition for the expression of their lethality. There is another aspect of lethals which we call as balanced lethals, and this is a beautiful expression of how things happen in nature to balance the good and the bad. These lethal genes, they are actually linked in a repulsion phase of linkage, and they are going to balance themselves in such a way that one of the good and one of the bad always stay together. So, that means if they are going to be maintained in repulsion phase due to very tight linkage, then the crossing over is going to be low, and therefore, the genetic recombination ratio is going to be less, and the recombination frequency being less, we would find that the recessive allele of one gene and the dominant allele of the other gene would remain predominantly on the same chromosome to balance. In other words, to explain this, if one parent has capital A or small b, the other parent would have small a and capital B. So, there is less of recombination frequency, the linkage is strong, and therefore, such combinations would continue, and they would try to ensure that the combination remains heterozygous. And if they are going to ensure that the heterozygosity is maintained, that means obviously the homozygotes would appear in less number. This is how the things are balanced. No discussion on lethality would be complete without the RH. However, we will not give much time to this, because this has been expressed in many other examples. The RH blood group actually is based on the presence of two genes, RHD and RHCE, which are located on the chromosome number 1, and to be precise on the smaller arm of chromosome number 1. Now, they are actually positioned just 31.8 kilobases apart, and we are also familiar with the idea that the RH plus is dominating over RH negative, but then here we are talking in terms of with reference to lethality. We would find that a child is going to receive RH plus gene from the father if the father is RH plus, and he starts producing RH negative blood. That is, the mother's blood is RH negative, and therefore, what is going to happen is that a marriage between the RH plus father and the RH negative mother would have a very high risk for the child, which we call as erythroblastosis fetalis, because some of the child's RH plus antigens are going to travel to the placenta to the mother's blood, and the mother's blood is going to start producing antibodies which will attack the hostile antigens of the baby. And if the mother had previous RH plus pregnancies, those antibodies would still be persistent, or if there was a transfusion with RH plus blood. In both the cases, the antibodies against RH plus would always be present in sufficient numbers, and therefore, these antibodies from the mother, they would enter the child and would start destroying the blood cells, which would result in erythroblastosis fetalis. Finally, we have the fifth category, which is the gametic lethals. Now, this is also a very interesting aspect, because some genes are going to make the gametes incapable of fertilization, and there is going to be a complete change in the ratios, and such genes are called as gametic lethals. Here we introduce the concept of meiotic drive, and this concept tries to explain the gametic lethals. Meiotic drive has been defined as a possible mechanism which leads to the production of unequal number of functional gametes by a heterozygote. In normal Mendelian genetics, we have learned that each heterozygote is going to produce equal number of gametes. Now, there are certain males in Drosophila, a species being Drosophila pseudo obscura, which produces only half the amount of sperms as compared to the normal males. Now, how does it happen? When these males are mated to normal females, most of the progeny are females. Now, this demonstrates that the sperm cells which were produced by these males, they would be definitely containing the X chromosomes only, and their sperms having Y chromosomes are somehow non-functional. I may remind you that the Drosophila also shares the XX, XY type of sex determining mechanism. So, in this case, if the parents we have the XX as female and the XY as male, the gametes would be of, since the female is homogametic, only one type of gametes, that is X. But the male being heterogametic, two types of gametes are formed in equal proportions. However, due to a mutation, this Y chromosome happens to be non-functional. So, this gamete is useless, and therefore, more and more prevalence would be in the progeny of the females. So, this is how we have a gametic lethality, which is also supplemented by zygotic lethality, and which shows a balancing between the lethal systems. On our next turn, we would like to now pay attention more towards a different aspect. We always say that are the pleiotropic genes polygenes? No. We are going to elaborate upon this in the next lecture as to what are polygenes and what is the polygenic inheritance. And we are definitely going to reach a conclusion that many genes together are going to work in tandem and also cumulatively, perhaps additively, to give rise to a particular character. Thank you.